we're going to have a little bit of fun here. We're going to evaluate some drugs together. I hope you don't mind. So this is Ronald Fisher. Who here heard of Ronald Fisher? A few of you have. He's considered to be like a father of statistics, of statistical inference, or one of them anyway. That has a lot of fathers and few mothers. And he's also a mathematician, was a mathematician, and geneticist and biologist. And perhaps most famous for designing very, very careful agricultural experiments, such as this one. This is a design called Latin square design, where we're trying to evaluate how well certain crops work. If you notice, it has this 5x5 grid. And Fisher was pretty wise to the fact that you need to disentangle two sources of variability. So the crops will be 5 different types of crops goes down the column, but the soil condition change, right, down the rows. And so, you may have a little bit more sun up the top, and you may have a bit more moisture up to the bottom. And he thought a lot about how do you disambiguate these two different sources of variability. And him and his colleagues and later came up with a statistical framework that's based on things like p-values, which you sometimes see, and null hypothesis significance testing to essentially assess if there is sort of signal in the noise. Now, fast forward to today. This is a recent survey that was done in Nature, where they asked 1,500 scientists if there is significant replicability crisis. And over half said there is a significant replicability crisis. Something like 38% said there are some crises. And these kinds of results are actually analyzed. There are people who tried to do replications, and over half or something like that of all published research turns out to be false, which is kind of a bummer. It means if you pull out a research paper or out of a medical journal or any other, you can't just trust that the results there are true. That is a bit disappointing. So enter the age of clinical development. So we generally, and what I'm going to discuss is the clinical part. So we work in phase 1, phase 2. But this is where some of the methods that were developed by Fisher and his colleagues continue to be used today, and he would probably turn in his grave if he knew just how they're being used. But for example, in late-stage clinical development, it seems like these methods are fairly safe, where you have randomization, you have lots and lots of patients. A late-stage cardiology trial may have 10,000 patients or something like that. They're randomized to different arms and so on. So you might think, well, that's fine. We should be able to actually detect something that's real. But a thing that happens there is that you often reach statistical significance and the drug gets passed, but there's no clinical significance. In other words, the drug doesn't make anything, doesn't actually improve patients' lives. Case in point, I'm not going to name the drug, but there's one that was recently approved for Alzheimer's disease that essentially showed, and don't quote me on this, but something like the effect size was like less than 1 on a 30-point cognitive score. But it reached statistical significance. It was approved. Now a few people from FDA resigned, and it was a big scandal. What's maybe a bit more upsetting, that similar methods are used in the early stages, where phase 1, phase 2, there is no randomization. And so I'll give you an example. If you go to SITC or ASCO, one of these oncology conferences where they release early results, you may see a picture like this, where you're looking at, let's say, just look at the clinical benefit rate in panel A, right? You have this green drug and you have this purple drug, and the response rate. So here, we're just saying, well, how many people responded? In other words, they given these people a drug, and they measured their tumors and how many shrank them by some percentage point. An extremely coarse-grained measure of efficacy, not very adequate. It says here that they report a p-value, which is kind of a ridiculous thing to compute for this kind of thing, and they would say, well, nothing is going on, it's probably a bad drug. It may be a bad drug. It may be not a bad drug. I don't know. It's not enough for me to really tell what's going on from this picture. Okay, how about this one? Suppose this is what we actually often see. Let's say we have this drug that's being compared in early trial, 30 patients in each arm. The purple one, 5 responders. The green one, 15 responders out of 30. How many think it's a great drug? Raise a hand if you think it's a good drug. Can't really see you. I actually don't know if it's a good drug. The chances are, if you're seeing this, it's probably noise. How can this be? Well, 2 things. The sample size is very small, 30 patients. Out of a small sample, you can observe something like this just by chance. No, that's number 1. Number 2, if it were published, it's probably publication bias. People don't publish results that are not so good. They typically publish results that are good. So if you're actually seeing this being published in this sample size, the chances are nothing is going on. Lots of biotech companies went public on results like this. It didn't turn out so well. And often you see something like this in an early trial. You don't even have a comparator, right? There's no purple. You just have an active treatment arm. 15 people respond. Is that a great drug? I don't know. I can't really tell. Okay. Now we enter the world of precision medicine. What is precision medicine? Precision medicine is supposed to treat a very specific subset of a population. By the way, you ask today a physician, he'll tell you he practices precision medicine, right? He's treating that person right in front of him. But that's not exactly what we're talking about. What we're talking about is designing drugs that target very specific subset. If this will come to pass, your late-stage trials that today have thousands of patients are going to look like your early trials that have 20 or 30 patients. So in that setting, the problems that you're seeing are just going to be exacerbated. Not great news. Okay, so I'm telling you about problems, right? Like statisticians are grumpy people. They're like, they like to talk about why things don't work. So you're in luck. I'm not a grumpy. I'm a statistician, but I'm not so grumpy. So I'll tell you about what are some of the things we can do today, even with the molecules that we have, to do better. So the first thing is that this is an example of a patient whose tumors were measured over time. So the blue line is measurement at baseline was the first, and then you can see like the patient is responding. As you can tell from this picture, there's a lot more going on than just whether somebody responded or not. So when you cannot enroll enough patients, okay, that means you need to measure more meaningful things on each individual patient. That's, in fact, what we do. We have a tumor growth model that predicts how each individual patient disease will evolve over time. And from there, you can then compute things like how durable is this response, how deep is the response, other things that are very, very important in differentiating the treatment than just whether somebody shrunk tumors by some percentage point. So point 1, we need good biomarkers and we need good longitudinal models of the disease. Point 2, this sharply dressed guy, Laplace, lived 200 years before Fisher, okay? And he used something called Bayesian inference, although at the time it wasn't called that. And that gives us something that we actually want. It computes the probability of the effect being true, because that is ultimately what we want. We're not interested in p-values or other kind of measures that are very difficult to understand and generally not so good at differentiating between treatments. We want to just know what is the probability that things are working. If this is new to you and you'd like to learn, there's a terrific book called The Theory That Would Not Die, How Bayes' Rule Cracked the Enigma Code, Hunted Down Russian Submarines, and so on. Highly recommended. So that's number 2. Number 3, if you cannot enroll another patient and you cannot measure anything else on this particular patient, what are you going to do? Well, you have to bring in external data sets. That's your third prong, right? That has to do with creating synthetic controls. In fact, we do that a lot. But there's a big caveat here, which actually I'm going to give a hat tip to April and Michael, I think, from NIH, that talked about really bad biased data in real-world evidence data sets. So how you collect this data is very important. We only use, our preference is to use clinical trial data from completed clinical trials in related diseases. If we don't have that, we use some of the data from real-world evidence, but it's not, it's patient records and not things like claims or billing codes, which are not very useful. Even there, we have to do a lot of work to deconfound this data, okay? And to do that, there's a well-known framework that has to do with, and it's called causal inference, or the sort of science of cause and effect, as Judea Pearl calls it. So I'll give you another book recommendation if you like. It's called The Book of Why, terrific exposition on the causal inference, particularly why it's extremely important in the age of AI. Okay, so that's number 3. Number 4, the way that the trials are actually designed and run is not very optimal because you look at the large clinical trial, you enroll 1,000 people, you wait 5 years, and then at the end you say, did it work or didn't? I think that's not very ethical, actually. Some of the best trials that are being designed today are adaptive. There's adaptive randomization. We direct more people towards treatments that are working. And if you want to learn more about that, there is a fairly successful platform trial for breast cancer called I-SPY 2. You can go check it out. So adaptive trials is what I hope what the future holds in terms of trial design. And last but not least, of course, you should talk to your friendly neighborhood regulator, which would be FDA here in the US, or if you're in Europe, the EMA. And we do talk to them. They don't invite us to their house for dinner, but we do have occasional meetings. And I'm actually surprised that they're very open-minded and very much know what's going on, what's coming. But we need to do kind of more to push them along. To help to move the regulatory framework forward. And that is all I have for you. Hope you enjoy the talk. As the kids say, click like and subscribe. Feel free to hit me up on LinkedIn, send me an email. Hope you enjoyed the talk, and best of luck today and during the rest of the HitLab Summit here in New York. Thank you.